Hello, welcome to the channel, guys. Today we're going to discuss about the complement system. Okay, so diving into the topic, I want to make some introductory part of this complement system. So, complement system is part of immune system which involves certain proteins, multiple proteins. Okay, these proteins are present in blood and mainly present in inactive form. When some pathogen are partially encountered, this complement system help the immune system to completely kill that pathogen. So as this system complements the immune system, this system is known as complement system. Okay, so next, so let's move forward. Okay, so as I have already discussed, complement system is a part of immune system. Okay, it is part of immune system which involve certain proteins that work together to fight and clear pathogen. Okay, there are mainly there are more than 20 types of proteins present in complement system. Okay, these proteins are generally inactive in blood and get activated in response to microbial invasion or injury. When when a person get injury or some pathogen invade in its body or in its cell, these proteins are get activated. Okay, once these pathogen are activated, they initiate a cascade of reaction which help the immune response which help in immune response. What is cascade of reaction? Cascade of reaction means a series of reaction taking place one after another. Okay, so after the cascade of reaction, they form a membrane attack complex. Okay, they form a membrane attack complex by aggregation of complement proteins. Okay, complement proteins are generally known as C proteins. Okay, so what are the functions of complement system? There are mainly three functions of complement system. There can be more than three, but there are mainly three function of complement system. The first thing is opsonization. Okay, so let's understand the opsonization process. Okay, some let's suppose this is a pathogen. Okay, this is a pathogen. So complement proteins, what complement protein will do, they coat this pathogen. Okay, they coat this pathogen, and this coating will increase the appetite of macrophage. Okay, and macrophage can easily engulf this pathogen. Okay, this it is called opsonization. Okay, so next thing is chemotaxis. What is this chemotaxis? Okay, let's understand this with diagrams. So it is a pathogen which is present very far from the macrophage. It is macrophage present here. This is very far. Okay, so complement proteins will coat this pathogen, and these release some chemicals which attract this macrophage towards the pathogen, and now this macrophage will engulf this pathogen. Okay, next is cell lysis. Okay, notice that in these two processes, in these two functions, first involved macrophages. Okay, and second, immune cells are involved. In third function, immune cells are not involved directly. This is done by the complement proteins. Okay, what they will do, okay, this is a pathogen, and complement protein will surround this pathogen and form spore in the membrane of pathogen. Okay, so after forming pore, all the contents of this pathogen will come out. Okay, and as it come out, the osmoregulation will be imbalanced. Due to the imbalance in osmoregulation, the pathogen will die, and this is how the complement system directly kills the pathogen without involvement of immune cells or macrophages. Okay, and another function can be inflammation. Okay, inflammation. Okay, so let's move forward. We already know that complement proteins are generally inactive. For activation of this complement proteins, there are three pathways. There are other pathways, but three pathways are generally most important. The first one is classical pathway, the second is alternative pathway, and third is lectin pathway. Okay, so for more information, I am telling this. The alternative pathway is part of innate system. Okay, innate system. After the alternative pathway, the pathway takes place, and after these two, the classical pathway will takes place, and it is part of humoral response. Okay, humoral response means it requires antibodies for its activation. Okay, one by one, I will discuss about these three pathway. So first, we'll discuss about the classical pathway. Okay, so let's discuss about the classical pathway. As we discussed in previous slide, that classical pathway is a humoral mediated pathway, means it requires antibodies. It requires antibodies for its activation. Okay, so in classical pathway, there are lots of protein involved. These protein are named as C proteins. So from C1 to C9, nine complement proteins are involved in classical pathway. Okay, so complement proteins are made up of two subunits, Ca and Cb. So generally Ca are smaller subunit and are part of anaphylotoxin. Okay, but Cb are larger subunit, and these subunit bound to the pathogen and complements the immune system. Okay, so these Ca part are generally degraded, and Cb part are generally bound to the pathogen. Okay, here mentioned that Ca are anaphylotoxins and are generally released or degraded, and Cb are large subunit and binds to the antibody. Okay, so at first C1, the complement protein 1 will come and bind to the Fc region of antibodies and recruit C4. Okay, let's see this. So it is a pathogen. Okay, it is a pathogen. Now some antibodies will attack this pathogen, and the first complement protein is C1 protein, which come in place and bound to the Fc region of antibodies. And this C1 recruits another protein, that is C4 protein. Okay, C4 protein, and degrade this into C4a and C4b, among which C4b will bound to this uh, pathogen, and C4a will be degraded. Okay, after this, all nine proteins, okay, C3 to C9 protein will become the part of cascade reaction and form membrane attack complex. What is membrane attack complex? Membrane attack complex means a group of protein which form a complex and makes pour into the membrane of pathogen and kill the pathogen. Okay, so let's move to the next slide. As I have discussed that the complement proteins are made up of two subunit, one larger and one smaller. Generally, the CB are larger subunit, but in case of C2, okay, in case of C2, the larger subunit will be C2A, and the smaller subunit will be C2B. Okay, but in all cases, the larger subunit will be CB part. Okay, so the cascade reaction, this cascade reaction involves all the nine complement proteins in order of first the C1, second C4b, third C2A, fourth C3b, C5b, C6, C7, C8, and then up at last C9, which form the membrane attack complex and makes pore in the membrane. Okay, clear? Now let's understand about the this cascade reaction with a diagram. Okay, see, at the beginning, a pathogen is entering in the a pathogen is entering in the body. This pathogen have an have an antigen. Okay, this yellow thing is the antigenic part. This antigenic part is attacked by these antibodies. These green are antibodies. Okay, antibodies, and this the Fc region of antibodies are bound by the C1 complex. Okay, it's the C1 complex. This C1 complex recruits C4 and and uh, degrade that into C4A and C4B. Only C4B will be attached to the C1 complex and C4A will be degraded. Okay, after this, the C4B act as C2 convertase and breaks down C2 protein into C2A and C2B, among which C2A, the larger subunit, will attach to the pathogen and C2B will be degraded. This complex of C2A, C2A and C4B act as C3 convertase, and this C3 convertase convert the C3 into C3A and C3B, among which only C3B bound to the pathogen and C3A released. Okay, in next these three proteins, that is C2A, C4B, okay, C2A, C4B, and C3 act as C5 convertase and degrade the C5 into C5A and C5B. Now C5 will be degraded and C5B will bound to the pathogen. This C5A, C4, C3, and C2A act as sorry, this C2, C2A, C4B, C3B, and C5 recruits other three proteins that are C6, C7, and C8. Okay, and these three proteins recruits C9. Once the C9 bound to the pathogen, they form membrane attack complex and makes pore in the membrane and kill the pathogen. Okay, it is the classical pathway of complement activation. In next video, we'll discuss about the alternative pathway and lectin pathway of complement activation. Thank you.